Hello everyone, in this video I'm going to be talking about the action and absorption spectra of photosynthesis. Now it's important to understand the difference between the two and what they show in relation to the pigments involved in photosynthesis and the way in that plants are adapted to maximise the process. So let's start first of all with the action spectrum. So I'm going to just draw a graph here, and on the bottom we're going to have wavelength. So we're looking at the action spectrum for this. So we've got wavelength in units nm, so nanometers. We're going to have 400 in this region here, so we'll start at 400, then we'll move to 500, 600, 700 here. So what I'm going to do is build up the action spectrum because it shows you the photosynthesis rate in relation to the different wavelengths of light that plants absorb. So the 400 to about 520 region, this area here, is the sort of violet range. So I'm going to colour this in purple here. This is the violet range from about 520 to about 630 or so. This is the sort of green-yellow region. So we've got the green-yellow wavelengths of light in that bar here. And then when we're looking at 700 nanometers, that's verging then into our sort of, we're looking there at more orange to red end of the spectrum. So if, like I said, we had photosynthesis rate, at the top here, and put that as a percentage, I think, for units, then what you get is a very characteristic graph. So we'll just put in the scale here, 20, 40, 60, 80, and 100. It's not the best scale I've ever drawn, but you find something very distinctive. It's a bit like this. It's quite an unusual shaped graph. Now this is called the action spectra. So let's just make that clear. This is the action spectra, and it shows that the violet and actually the blue light is used more efficiently by the plants for photosynthesis. Green light less so. Now the shorter the wavelength of the light, what we can see here if we're looking at the violet end, that means that those photons of light that we need in photosynthesis have greater energy, and the greater the energy, the more chance that those electrons in the chlorophyll pigment will be excited to a higher energy state, a higher energy level. And that's critical in the first steps of photosynthesis. So the plant is able to make use of the violet end and even the longer wavelengths of light. So we talk about 700 here. We can see to a degree the plants are also able to make use of that. But we have this unusual part here. It's where I'm drawing the arrow. Now, my students ask me why that graph doesn't hit the x-axis. Because when we look at, say, green leaves, you're taught that those would absorb all the colours of white light but reflect back the green. So it's as if they're not using the green part of the spectrum. But in this region, this graph would imply that even in the green spectrum, there is still a value for the photosynthesis rate. So the plant somehow is actually using some of that green light. And that's where the absorption, this is where the absorption spectra comes in. Because what the absorption spectrum does is show you the percentage absorption of the light in relation to the different pigments that you actually have in the leaf at the given wavelengths. So I'm going to draw a second graph here, and this one is the absorption spectra. Now this graph has exactly the same x-axis. So on the bottom we have wavelength of light in nanometers. But this time on the y-axis we have absorption as a percentage. And again we'll have 0 and we'll go 20, 40, 60, 80, 100. Now this graph looks a little bit more unusual because there's more than just one line. We talk about chlorophyll in the plant, but in truth we don't just have one type of chlorophyll. In fact, I'm going to draw the different types we have. So for starters, we have one that looks a little bit like this. Now that one there refers to, in fact I'll use a colour because I've already used the blue, refers to what's called chlorophyll A. So we have chlorophyll type A, but there is also another type, chlorophyll type B. Now chlorophyll type B has a slightly different curve. So we'll label that one chlorophyll type B. So we have chlorophyll A and chlorophyll B, but there is another one that we can include onto this graph, and I'll draw this one in a blue colour, I think, and this one is for something known as carotenoids. So the graph looks a little bit like this. It's quite unusual. There we have it, and we'll label that one carotenoids. So here we have our absorption spectra. Now, ultimately, what this graph or this spectra is showing is that at different wavelengths of light, we have different percentage absorptions of sunlight in a plant, or in a leaf more specifically. So we can see that in the 400 to 500, very short wavelength range of light, we can see that we're getting quite high absorption. If we look at the red peak, if you like, quite a high amount of absorption there, and that correlates with what we said in the Action Spectra. Then in the 400 to 500 wavelength area, the rate of photosynthesis is quite high. So I'm referring to this region here and this peak here. We can also see on the absorption spectra that we have an equivalent peak at around 700 nanometers. But what's interesting is the sort of 520 to 620 nanometer wavelength range. Because I said previously on the Action Spectrum that we can still get some of that green light being used. The rate of photosynthesis wasn't at zero, even though the leaves typically appear green and as if to be reflecting out that light. But when we look at the absorption spectra, we can see why that rate of photosynthesis isn't zero. It is because we are still getting some absorption. If we look at this point here where I just put an asterisk, at that point we are still getting some pigment absorbing some of that wavelength of light. So what we have in the chloroplast isn't just chlorophyll, well, just one type of chlorophyll. We've got chlorophyll A, chlorophyll B, but we also have what are known as accessory pigments. Now these accessory pigments, I'll just put that here, accessory pigments, such as the carotenoids, and also a group called the xanthophylls. That's Xanthophylls, xanthophylls. The xanthophylls and carotenoids act as accessory pigments and absorb wavelengths that chlorophyll can't. So the whole purpose of this really is to maximise how much sunlight that plant can use productively for photosynthesis. So in this video I've just talked about the difference between the action and absorption spectra. I just want to very quickly just say a little bit about the story behind it and where this idea comes from. In 1881 there was a German plant physiologist called T.W. Engelmann, and what he did was place a filamentous green alga under the microscope, and he illuminated it with just a tiny spectrum of visible light. And in the medium surrounding the strands were motile aerobic bacteria. And what he had noticed was that after a few minutes, the bacteria had congregated around the portions of the filament illuminated by red and blue light. And he assumed that the bacteria were congregating in regions where oxygen was being evolved in photosynthesis. So even back then in 1881, Engelmann had concluded that the red and blue light were the most effective colours, if you like, for photosynthesis. So what we have here in the action and absorption spectra really is just a development, if you like, of Engelmann's work, and just showing how the rate of photosynthesis and absorption percentage vary with the different wavelengths of light that we have. Okay, hope all that helps.